relentless curiosity, the pursuit of knowledge, the search for advances, the drive to overcome challenges and find answers. These are what fuels research at the University of Chicago and other leading academic research institutions around the globe. Today's world is filled with extraordinary opportunities and potentially existential threats, from quantum science and AI to climate systems engineering and sustainable water recovery technologies to new math models of biological processes. The research endeavors undertaken at UChicago hold the potential to significantly shape our future. But what goes on behind the scenes of this groundbreaking work? And why is understanding this research so vital to our collective success? In the UChicago series, Inside the Lab, you'll experience this innovative research firsthand. Go behind the scenes, meet the researchers, and hear how their work could help solve some of the world's most complex problems and deliver upon the biggest opportunities of our time. From small research groups to large national labs, scholars are testing theories, venturing into uncharted territories, and pushing boundaries across diverse fields. Come explore Inside the Lab, where UChicago's researchers are building our tomorrow.
UChicago scientist Xuan He and his lab are investigating how things that happen in cells can help us address global hunger and even adapt to climate change. Find out how inside the lab. In 2011, Xuan He's lab made a surprising discovery about how cells work. Cells contain DNA, which is like a set of instructions for how living things carry out their lives. DNA's cousin, RNA, has a crucial role to play in how those instructions are carried out. He's lab discovered that a process called RNA methylation controls which genes are turned on and off. The lab used this to affect gene expression in plants, which could have a huge impact on crops.
Animals, insects, or even bacteria can run away. Plants, unfortunately, have to stay where they are. So what that means, plant has a complicated system to respond to the environment.
Normally, if a plant is stressed, it uses methylation to grow more slowly, but He's lab found they could turn that off, which makes the plant grow faster. Early tests suggest that this experimental method could boost the yield of crops by as much as 50%.
The ability to combine high yield with drought resistance is going to be amazing.
In the lab, the plants grow longer roots, also making them more resistant to drought and extreme temperature. The He lab's discoveries could help us adapt to a changing climate and address global hunger.
When you work on basic science, you come up with unique discoveries. There are many avenues for potential real-world applications you can impact the society.
Discoveries like these benefit all of us, and they can only happen inside the lab. At the Institute for the Study of Ancient Cultures, or ISAC, a team of UChicago conservators diligently preserves some of the world's oldest objects. Find out how inside the lab. A lot can happen to archaeological items over time. Light can fade colors, water can corrode. Heat, insects, and human conflicts are constant threats to the materials that help us understand the past. For many objects, ISAC's conservation lab is the last defense before they're lost forever.
In the broadest sense, a conservator is responsible for the preservation of a collection of cultural heritage.
ISAC exhibits, preserves, and cares for thousands of ancient objects. The Conservation Lab is currently preserving part of a temple wall built over 2,500 years ago. The collection of 282 glazed bricks once formed part of the Sin Temple in Dur Shurukin, an ancient Assyrian city located in modern-day Iraq. In 1931, archaeologists excavated the bricks and packed them into 35 crates bound for Chicago. Nearly 100 years later, conservators are carefully unpacking them brick by brick. Each brick is moved, cleaned, and stabilized. Adhesives are used to fit fragments back together like thousands of puzzle pieces. As the bricks are conserved, figures emerge, including a bull, an eagle, and a lion.
The fact that we can actually recreate this frieze is amazing. This is an entire facade. Even with all the damage, it's still intact.
The team hopes to display the completed facade in the ISAC Museum.
We're all in this business because we care about cultural heritage and we want to maintain it for the next generation.
The work of conservators helps us make connections to the past to better inform our future. And it can only happen inside the lab.
Human lungs take over 8 million breaths a year, but they are constantly threatened by pollution, viruses, and cancer. Joyce Chin's lab at UChicago's Pritzker School of Molecular Engineering is pioneering ways to understand lung disease at its earliest stages. Find out how inside the lab.
Lungs are complex organs that are directly exposed to the outside environment. But studying disease in human lungs is very challenging.
Human lung is a beautiful organ, yet complex and difficult to study. Because of the complexities, multi-disease can happen in different locations of the lung system.
To develop treatments, scientists need to know exactly how these conditions progress, starting at the beginning. But we often don't catch lung diseases like cancer until it's too late.
Small cell lung cancer is the most aggressive type of the lung cancer and one of the most aggressive type of the cancers in human body, along with pancreatic cancer.
Chin's lab specializes in developing innovative ways to recreate lung cells in the lab so they can study what happens at the first stages of disease. They create special stem cells from skin, blood, or hair cells instead of embryos.
The goal is we want to create multicellular structures and to study these ecosystems in human disease.
From there, they can create miniature versions of lung structures in a petri dish to understand how disease begins and spreads. They can use these in experiments to understand the lung as a whole system, how the lung responds to threats like pathogens or fungi, what genes are involved, and whether we could regenerate lungs after they've been damaged. This kind of research is innovative and painstaking, but could help address diseases that affect people around the world. And it can only happen inside the lab.
Professor Jens Ludwig and the University of Chicago Education Lab design, test, and scale programs to set up students for future success. Their innovative research is helping students nationwide overcome pandemic learning loss. Find out how inside the lab. COVID-19 created not only a once-in-a-century crisis in public health, but also in our schools. In 2022, a national report on education documented the largest decline in national reading test scores since 1990 and the first decline in national math test scores ever recorded. But these declines weren't evenly distributed. Students who were already furthest behind were the most affected by pandemic learning loss. This historic learning drop could set an entire generation of students off track for the rest of their lives.
This is one of the biggest issues that we're not talking nearly enough about. And there's good news here. There's a solution to this.
The Education Lab's solution is high-dosage tutoring, small group tutoring for 30 to 45 minutes delivered during the school day. The Education Lab, in partnership with the Chicago Public Schools and the nonprofit Saga Education, delivered this intensive tutoring to thousands of students beginning in 2013. The results showed that this intensive tutoring doubled and sometimes tripled what students learned in 1 year's time.
The problem is not that we don't know what to do. We just need more resources to help schools to solve this problem.
To scale tutoring to students across the country, the Education Lab is figuring out what types of tutoring help each student learn as much as possible at the lowest cost.
We have been trying ways to selectively incorporate technology to bring down costs and bring down the number of tutors that you need.
To do this, they're partnering with school districts around the country to co-design locally tailored variations of tutoring, rigorously measure impacts on student learning, and help district partners scale the versions that work best for their students. The ultimate goal is to create the blueprint for every district in the U.S. to help as many children as possible, as effectively as possible. And it can only happen inside the lab.
Hundreds of species of microorganisms live in our intestines. Known as the gut microbiome, these bacteria, fungi, and other microbes are mostly beneficial, but we're only beginning to understand how important they are to our health. Microbiologist Sam Light's lab in UChicago's Department of Microbiology is investigating ways to harness the gut microbiome to treat and cure disease like cancer, diabetes, and even Alzheimer's. Find out how inside the lab. After we swallow food, our digestive system breaks down and extracts energy and nutrients. Our gut microbiome breaks down what's left over and produces molecules known as metabolites, which can behave in similar ways to medicine.
They're small molecules, they can enter our bloodstream, and then they can modulate like different biological processes, and that can have like a variety of different implications for human health.
In his lab, Light and his fellow researchers collect samples that can be used to grow microbes and extract metabolites for studying. They grow these microbes using specialized vacuum chambers that mimic the oxygen-free environment of our large intestine. This ensures that the microbes perform the natural fermentation process that happens in the gut microbiome. The metabolites are then separated and analyzed using mass spectrometry machines. Then they are cataloged to determine how they contribute to health or disease.
We're still kind of like at a very early stage, in my opinion, of like understanding the microbiome, like what all of these different microbes are doing, kind of how they interact with us.
Light's lab is untangling these complex relationships so that we can develop a fuller understanding of how the gut microbiome can create new treatments and perhaps cures for disease. And it can only happen inside the lab. Our brains contain over 400 miles of blood vessels. But when these blood vessels fail, it means serious trouble. Neurosurgeon Issam Awad's lab at the University of Chicago Medicine is working to find earlier warning signs for those most at risk for stroke and bleeding in their brain. Find out how inside the lab. Stroke and brain bleeds are among the leading causes of disability and death worldwide. Some strokes happen after blood vessels in the brain weaken and rupture, but it can be difficult for doctors to predict why and when this might happen.
I deal with people who are now having to go to rehab because their brain is so damaged. That is the tragedy.
Awad's lab researches the science behind why brain bleeds and strokes happen and studies ways to prevent them.
I'm a brain surgeon, and the other half of my life that is devoted to research is to try to understand why this happens. And we have to be able to tell the person you are at much higher risk, and we can monitor that risk and modify that risk.
Awad's lab developed specialized MRI scans that measure iron in the brain. The amount of iron tells scientists how much bleeding has occurred. The team also created imaging techniques to detect smaller bleeds and vascular leaks that could be precursors to larger hemorrhages. They also measure molecules in the blood and analyze them to search for signals that can show whether a person is at risk,
We have identified some signals that actually predict a bleed in the following year, so that you would be able to know that the blood vessel is weakening but hasn't yet gotten to the point of bleeding.
The research could also lead to new medicines, which could help stabilize blood vessels and prevent strokes from happening in the first place. And it can only happen inside the lab.